Hello, my lovely anatomist and physiologist, Michele Glass here, and we're going to revisit something we've talked about before. Now, repetition is always good when we're learning, especially when we're learning something new, especially when we're learning something complicated. So what I want to revisit is this idea of the systemic circuit. So let's start with our heart. And from our heart, remember, the blood is going to move into our arteries. And so we spent a whole conversation on arteries. We talked about arteries always carry blood away from the heart. We say they have a very thick wall. We said that they have some elasticity and they have a really high blood pressure. From the arteries, we move into these smaller vessels called arterioles. So from the arteries, we move into these smaller arteries, basically called arterioles. And what we see is when we talk about them as being smaller, the lumen, which is that inner space, the lumen has a diameter of about 30 micrometers. So this is going to be much smaller. This is going to slow down your blood flow. So we're going from a bigger tube to a smaller tube. So we're slowing down our blood flow. And we're going to see that there's, like, as a standard operating procedure, a slight constriction to your arteriole. There's a slight constriction to your arteriole. And this is going to be super important in helping to regulate blood pressure. Super important in regulating blood pressure and blood distribution. So hopefully you have this idea already from our study of the autonomic nervous system that we can redistribute our blood flow as needed. So if we're doing more physical activity of our body, meaning like our skeletal muscle and movement that way, we need the blood to flow to our extremities and to that muscle. If we're resting after dinner, watching some Netflix or whatever, then we need that blood to flow instead to our digestive tract to help with that absorption, digestion, and absorption of nutrients. So we'll see that instead. Remember, when we're talking about this kind of control, the slight constriction and so forth, we're thinking really the sympathetic nervous system as really being in charge of controlling the diameter of our vessels. And here we're really talking about like muscle tone, right? We're talking about our background level of stimulation. Our default, our normal is to see that these vessels are always slightly constricted, okay? From there, we're going to move potentially to our capillary. Now we're going to see that there are, let me pull it up here a little bit more. We're going to see that there are 3 different types of capillaries. And so we're going to talk about those in a little bit more detail, in a lot more detail, actually, in their own video. Our diameter here is getting much smaller. So we've already talked about the diameter of a capillary before. I said it could be 4 micrometers. So let's go to that size, 4 to 10 micrometers. Our OpenStax textbook says 5 to 10. So you might see some difference there. I've put my capillary in red color, right? Because this is where exchange is happening. So if we're talking about our gases, oxygen gas will leave the blood and move to our tissues, whereas the carbon dioxide gas will be picked up. This is where we're going to see our nutrients leaving. This is where our hormones might leave or come in. And so we'll see all of this kind of exchange. This is one of our locations where we can get diapetesis, which remember is our term to talk about those white blood cells that can actually like squeeze in between the cells of the capillary and go out into circulation. We're going to see these are super tiny vessels, super tiny vessels. We're going to see that they are a basement membrane. We're going to see that they are endothelial cells. Y'all, that's simple squamous. That's a single layer of cells, right? And then we have sometimes, depending, so we can say occasional muscle cells here. And from the capillary, we move next into a venule. So a venule is sort of like analogous to our arteriole, meaning that it's a small version of a vein. And when we talk about it being small, we're going to see it has a lumen or diameter of 8 to about 100. So there's a lot of variation there, 8 to 100 micrometers. Now what we'll see is the venule will join many capillaries. So we're seeing quite a lot of blood then flowing into the venule because you've moved the blood from the arteriole to a lot of capillaries, and then several or a lot of those capillaries are feeding into the venule. We'll see also with the venule that we do get some diapetesis occurring here as well. So remember, when we talk about diapetesis, we're talking about those white blood cells leaving the circulation, moving into the lymph, moving into your body tissues to be like really effective at, getting rid of pathogens and all of that. Now, here's something that's really interesting. Here's something that's really, really interesting. Those capillaries are like, I'm going to label them here as resource allocation masters. And we're going to look at the capillary bed in more detail here in just on the next video, right? Just in a little bit. And what I mean here is, we are seeing blood flowing to all of our tissues in order to keep it alive. So all of our cells are needing some level of oxygen, some level of nutrition, and some level of waste disposal all the time. But our tissues are not equally active, and so they don't have equal needs, right? And so what we see with the capillary is that it has local mechanisms to know whether that tissue actually needs blood flow at that time. So when we talk about the systemic circuit and we say the blood flows from the heart to the arteries, to the arterioles, to the capillary, to the venule, that's not actually always true. We'll see sometimes blood will flow from the artery directly to the venule. And in that case, it's moving through what's called, and we'll see this on a picture, on a diagram in our next video, a met-arteriole. So sometimes we have this method, and it's up to the capillary to move blood directly from the arterioles to the venules. I think this is a very interesting, very important feature that we see. From the venule, then, we're going to see the blood flowing to the vein. The vein, remember, is that larger, more irregular lumen. It's that thinner muscular wall, although it does have a thick tunica externa. We talked about the vein as having valves, so that's going to be an important feature. We talked about it as holding 64% of the blood in the body, and we talked about it having those special venous reserves, which we mentioned already as being located in the liver, the bone marrow, and the integumentary system. Another important thing to pay attention to here, one of the other reasons why we have 64% of the blood here is that we have fewer veins compared to venules. So just like you could have several capillaries dumping into a single venule, you're going to have several venules dumping into a single vein. And then remember, the vein is bringing that blood back to the heart. The other important feature to pay attention to here is obviously when we talked about blood leaving the heart, moving into the arteries, we can represent all of that as red to indicate it's oxygenated blood. So oxygenated blood is moving through the whole arterial system. That's arteries and arterioles. And then the capillary is where exchange can occur. So we might want to represent that with like a purple color. And then as the blood moves out of the capillary and into the venule, and from the venule into the vein, we see that this is the deoxygenated blood. It has already delivered oxygen and picked up carbon dioxide gas. Up next, let's look at some more detail of that capillary. So we want to see what are the three types. And we also want to see like, ooh, how is this capillary such an excellent resource allocation master? So stay tuned. As always, take care of yourselves and each other.